first and foremost, I consider myself a neuroscientist. I study the brain, I'm fascinated about the brain, and that's what I think about basically all the time. I hope to convince you as well that the situation of blindness and visual impairment is an incredible model to study the brain and development. It's a really fascinating opportunity to understand how the brain develops. And that's strictly, obviously, from a neuroscience standpoint. I'm also an optometrist. I see a lot of patients, particularly with brain damage, visual impairments, children as well as adults. And my thought is that what I learn in the research lab can ultimately translate into the clinical care and education of these individuals. I'm also a big believer of assistive technology. I think that assistive technology, how ironic, huh? Technology is a good thing. There we go. I'm a big believer of technology, and I think technology is an enabler. And in my second lecture, I'll show you a little bit of work that we've been doing about video games and how we can use video games with blind children as a way to help with orientation, mobility, and navigation. And the final thing I'll mention is I also have a background in public health. I really think that what I do in the clinic and what I try to do in the research lab ultimately should translate in the way that we legislate and we take care of these individuals. And in Massachusetts, much like in Texas, these are states where we have invested in education. It's in the culture of the people, and I think that's really something extremely important. I think that's why things get done. So with that said, let me get started, and let me just give you the first slide to give you a sense of how I kind of approach these ideas and thinks about this aspect about neuroscience, cognition, behavior, and learning. If you're a neuroscientist, you fundamentally believe this mantra that the mind is what the brain does, right? That's really kind of what we think. And there is an intimate relationship between cognition, behavior, how we think, how we act, how we behave, and the structure and the function of the brain. How do we do this? Well, the first thing we do is we measure. We measure the brain in terms of its structure and its function as a response of somebody doing something. The second thing we do is we manipulate. We change the brain somehow and see how it affects cognition, behavior, and learning. That might be through natural causes like brain damage. It might be through a situation like visual development in the case, or development in the case of visual impairment. It might be through brain stimulation. For example, exogenous stimulation of the brain. I'll show you an example of that also. So this loop, if you will, allows us to create this understanding between how the brain acts, its structure and its function, and how we behave, how we think, how we learn, how we develop. Just out of curiosity, anybody know who this person is, the picture that I'm showing here? Bobby Fischer, yeah, the first American Grandmaster champion of chess. A very, very interesting individual. I always show his picture because if some of you know his history, he kind of had two lives. He was this incredible child prodigy, the first American to win. He beat all the world's best chess masters, and then after that became extremely recluse, had all sorts of psychiatric issues and paranoia and all sorts of things. And I always like this sort of picture to remind me just how quickly things can change, right, in a person's life. And if we understand the structure and the function, dramatically has impacts in terms of a person's life. So again, this mantra of the mind is what the brain does is fundamentally what we think about. Some things I always like to talk about when I get the chance to lecture with these groups. As neuroscientists, we're always kind of concerned about these neuromyths, these statements, if you will, that get propagated throughout cocktail parties and so on. I'm sure you've heard this one, this idea that we only use 10% of our brains. Yeah, just out of curiosity, who thinks that's true? I know it's being filmed, but don't worry, it's all right. You can tell me. we're friends, it's okay. Just out of curiosity, who thinks that we only use 10% of our brain? Anybody? You believe it? Yeah, you believe it? So also the person in the sound room believes it, yeah? I see that as well. Well, the reality is, and I think what you're trying to say is that we have potential, right? We can do much more than we think. And I think that's absolutely true, but I certainly don't think it's 10%. The fact of the matter is you use all your brain all the time. Doesn't matter how smart you think you are or who your favorite football team is, it's true. Everybody is the same. And here's the sort of the rationale. If you think you're only using 10%, by definition 10%, that means you know what 100% is, right? You have to know what the baseline is. What does 100% look like? It's like saying that person is only using 40% of their faith, for example. It just doesn't make any sense. You use all your brain all the time. What makes you different from me, from the person sitting next to you, is how it's wired, how it's connected. And I'm going to show you this examples of how blindness, in the particular case of blindness, and also in the case of CVI, are perfect examples of that principle. So that's the first thing to keep in mind. The second one that I hear a lot is 90% of all information that comes into our brain is visual. Same problem, right? If you know what 90% is, that also means that you know what 100% is. What is 100% information? As a scientist, I love to measure things. I would love to know what 100% information is. It's just that you just can't measure such a thing. At the same time, think of the counter argument. If 90% of our information that comes in is visual, does that mean that a blind child or a child born blind can only understand 10% of the world? This doesn't make any sense, right? So these are a lot of what I would say these sort of myths that sort of kind of get through, and I think they're important to kind of think about because, as I said, they tend to get sort of propagated. In fact, the only study I could find online from a peer-reviewed journal said that it's actually not. There's no evidence that it's 90%, to be perfectly honest with you. So I wanted to kind of share those points with you. So the main theme of my lecture and sort of the theme of the lab and what we do is this concept of neuroplasticity. So neuro obviously referring to the brain, and plasticity coming from the Greek word plastikos, which means to mold, right, or to shape. And just like a piece of plastic can change its shape and its form to do something else, so too can the brain under the right conditions. So I really like this definition. It's the ability of the brain to change its structural and functional organization in response to development, experience, the environment, or damage. This, in a nutshell, is what plasticity means. And you have this for life. It's an intrinsic property of the brain that you take with you throughout your lifetime. And there are all sorts of great stories. As you know, as neuroscientists, we always try to look at the media and so on and try to come up with some good neuroplasticity stories. So here's one I'd like to share with you, one of my favorites. This is a standard MRI image. You're looking in sagittal cut here. This is the person's eye. This is gray matter on the top here, white matter in between, and the little LV in the middle is the lateral ventricle. This is typically where fluid resides, cerebral spinal fluid. One day, this gentleman in France went to see his doctor. So I'm just showing you that's what a standard MRI image looks like of the brain. So one day this gentleman in France, and he was middle-aged, about 45, 50 years old, goes to see his doctor and says, you know what, I have these headaches, and they just don't seem to go away. And if you've lived in France like I have, I'm sure the doctor just told him, go home, have some wine, it'll be fine, don't worry about it, everything's okay. He comes back 2, 3 times, says, you know what, I don't understand, I've taken aspirin, I just don't understand what's going on. So finally the doctor says, okay, fine, we'll get an MRI. And here was the patient's MRI. That's his brain, right? About this 3-4 centimeter lip around there. And all that you see in black here is water. That's his cerebral spinal fluid. This guy had no idea that he was going through life like this. He got married, he got a job, he's a civil servant, he has 2 kids, has normal intelligence, had no idea that's what his brain was like until under the certain circumstances, quote, headaches. So it just goes to show you that something really, really important about the brain is that when things happen early and when things happen slowly, the brain is very, very good at adapting. When things happen very quickly and when we're adults, less so. Think about traumatic brain injury or concussions, for example. Very, very different scenario, and the brain is not as good as adapting under those situations. So just to give you a dramatic example of neuroplasticity. I also want to remind you that it's not a guaranteed fix. We have this idea that neuroplasticity can fix all our problems and don't worry, well, the brain will figure it out. Well, it's not entirely true. It's not a guaranteed fix. It's the inevitable consequence of how the brain works throughout a lifetime. And to give you an example of negative plasticity, I like this one. This is phantom limb pain, for example. So these are patients who have either a hand or an arm that was amputated or a leg, and they feel that pain, that their hand is in a vice. It's being crushed, right? Even though there's absolutely no limb. And the reason why is because the brain hasn't understood or doesn't adapt to the fact that limb is gone, and it signals that as pain, and massive chronic intractable pain in some cases as well. So plasticity, as I said, is the way that the brain adapts, but in some cases it can be negative and sometimes it can be positive. Our goal as neuroscientists, what we hope to do is to try to understand those cues. What makes it go one way versus another, and how do we leverage that in a positive way for rehabilitation and education. Another very, very important principle when it comes to brain development is the idea of the critical period. And a lot of the work that was done in this area were done by 2 of my heroes. This is David Hubel and Torsten Wiesel. They won the Nobel Prize in 1981 for their work. And what they did was they understood, essentially discovered and understood the physiology of the visual brain, the visual cortex. So some of the work that they've done, this is just one example, is they would take kittens and they would rear them under certain situations. They would suture one eye, they would close the other eye, change the different timings of that. They would put opaque lenses over the lens and over the eyes and so on, and they would get a sense of what the physiology and the anatomy of the visual cortex was. And that was really groundbreaking work, because from there, the way we take care of amblyopia, for example, is fundamentally a result of that work. So it's a wonderful example of how basic neuroscience research ultimately can translate into the care of individuals. And I think that's very, very much the strategy that we are all trying to pursue. Another thing to think about is that critical period has this sort of sense that it's finite, and it's really not true. We now know more and more that plasticity exists throughout a lifetime. It's certainly easier to learn a language when you're 6 compared to when you're 60, but it's not impossible. And I'll speak a little bit about that, and how, as we get older, why plasticity changes and why things get harder. But the fact of the matter is, plasticity is for life, and it's never, ever over. Okay, so a couple of questions I'd like to talk about specifically about the case of ocular blindness to set up the terrain, and then we'll switch to the case of CVI. So as I set it up this way, I think it'll make sense from a neuroscience perspective why we pursued this question first in terms of ocular blindness and how we got into the case of CVI. So a couple of things to think about. First and foremost, what are the behavioral changes in blind individuals? Are they better at non-visual tasks? So this is something I'm sure you've all heard about, and I'm going to share with you some scientific evidence that does indeed support that in the case of ocular blindness, these individuals do show superior non-visual performance. What about structural changes? Use it or lose it, right? How does the brain change as a function of the skills that you rely on? So as a blind individual who is a proficient Braille reader, do parts of the brain change as a function of that, for example, someone who doesn't? And I think a very, very interesting question are functional changes. So for example, what is the fate and the role of visual areas of the brain? How does the brain rewire itself in the context of profound visual deprivation? You all know that there's a large chunk of the brain dedicated to vision, right? So the question is, what happens to that part of the brain in the case of blindness? And we'll get into that as well. So here's a picture I'm sure you all recognize. This is Stevie Wonder, obviously who lost his eyesight at a very, very young age, was born blind, became a very, very successful artist and musician. I think there's a very interesting philosophical question you might want to ask yourself is, would Stevie Wonder be Stevie Wonder he is today if he wasn't born blind? In other words, did blindness somehow confer an advantage in terms of his ability to develop his musical skills and to be the person that he is today? And of course, it's not just in music. There are many, many arenas that we see very, very successful individuals who lost their sight early in life or later in life and have gone and had incredibly independent lives and successful lives. And I think it's an interesting question to ask, that what did blindness do in terms of that development? How did it actually impact them? So in terms of that question of enhanced compensatory behaviors, there is actually quite a bit of scientific evidence along these lines. There's evidence, for example, that individuals with ocular blindness have enhanced tactile perception. They also have enhanced auditory localization. They can identify sounds in space better than sighted individuals. Also better at identifying smell. Also better at verbal memory recall. And again, a lot of studies have shown this, so the evidence certainly seems to be there of these enhanced compensatory behaviors. But there are caveats, and I think that's an important thing to kind of think about. The first is what underlies these superior performances. First of all, it's not universal. It's not across the board. It's not every individual that shows this. The second thing to realize is that this is very much under very controlled scientific specific questions that we ask. And a lot of the times, these enhancements that we see in the lab don't necessarily translate to the real world. So that's an important thing to realize. Secondly, in terms of sensory thresholds versus attention, so what the evidence suggests is that this isn't enhanced hearing per se or enhanced mechanistic touch. It's enhanced attention spent or delivered to those senses when you take vision out of the equation. And again, we'll show you more evidence about that. Third point that's important to think about is comparison to sighted controls, right? So are the blind really better than sighted people, or are sighted people just really bad when you ask them to do this without their sight? I think it's an important way to look at it, both sides of the coin, if you will. And the last thing is contributing factors, the onset, for example, born blind versus later in life, the profoundness of the blindness, your education level. We don't know how these covariates intimately relate to these behavioral changes. I have a picture here in the top, in your top left, I should say, of Andrea Bocelli. I'll share a quick story with him. I had the pleasure to meet him a couple years ago doing some work for his foundation. And just to prove to you how big a nerd I am, when everybody was at the gala asking for his autograph and pictures and stuff like this, I went up to him and I said, Mr. Bocelli, do you hear better? Do you have a keener sense of touch? What's your verbal memory recall and all this? And he said, I've never been tested formally. I really don't think so. And when I asked him, why do you think you're the person you are today? And he says, it's really, really simple. I had an extremely dedicated family. I had resources early on. I never grew up thinking that I couldn't do the things that I wanted to do. It was all about resources. It was all about opportunities. You may know he was an individual who lost his sight when his early teens or around 10 or so. He was also a practicing lawyer for a couple of years, and one day decided, you know what, I I don't want to do this anymore. I want to be, a singer. That was my true passion. And sure enough, he's very successful today. So it just goes to show you that the neuroscience piece can only explain so much. At the end of the day, it's about resources and it's about opportunities. Okay, so here's that fundamental question that I talked about from a neuroscience perspective, the localization of function in the brain. And you all know this, that the 5 senses are all processed in different parts of the brain. Parts of the brain responsible for smell, for touch, for taste, for hearing, and vision. And the visual part of the brain in the back here is what's called the occipital visual cortex. And that's about 25 to 35% of your cortical surface. That's a lot of hardware dedicated for the purposes of seeing. So the question becomes, if you were born blind, what would all this do, right? Does it stay silent? Does it do something else? Can it do something else? If you're born blind versus losing your sight later in life, does that matter in terms of its developmental fate? So there's an interesting neuroscience question that a lot of people were interested in. And the way that we answered it was using a technique called functional magnetic resonance imaging. You're all familiar with MRI. fMRI sees the brain in action, and it works a little something like this. It turns out that when the brain works doing a particular task, the parts of the brain that are working the hardest are going to command more blood and are going to command more oxygen. And that sets up a contrast that the scanner can detect. So what we do is we ask an individual to do something in the scanner, look at pictures, count to 10 backwards, recall names, and then we scan the person doing nothing, at rest, if you will, and we subtract the two. And when we subtract the two, what's left over, these little hotspots of activity, theoretically are associated with the task that that person was performing, right? Everybody understand that? Okay, so let's look at some data. Here was the very, very first one. You're looking at the side view of a brain right here, and in this blue circle you see a large spot of activity in the occipital visual cortex of this individual, but this person is congenitally blind. So the question is why would their visual brain be active if they're congenitally blind? And the answer is this person is reading Braille. And that was one of the very, very first examples of these compensatory behaviors, that the occipital visual cortex is active for non-visual tasks. And many people have replicated this and also found in other scenarios as well, localizing sound, identifying smells, verbal memory recall, also in the case of language. In all these cases, the occipital visual cortex is involved in this processing. So the take-home message here is that the seat of compensation, or compensatory behaviors, I should say, in the case of ocular blindness, is the visual cortex. Turning your visual cortex into a non-visual processor, if you will, is part and parcel part to the compensatory behaviors in this group. Now I'm going to show you some more evidence that this is indeed seems to be the case. First question you may ask is that if I'm born blind versus losing my sight later in life, how does that change? Well, someone did look at that point. This is Harold Burton's work a few years ago. He found individuals who were early blind, lost their sight before the age of 2 or 3, versus individuals who he classified as late blind, lost their sight in their teens or in their 20s, who were matched for Braille reading ability. And that's the important thing. So they had the same Braille reading skill level. And what he found is that in both cases, the occipital visual cortex was active, but notice in the case of early blindness, there's a lot more activation than there is in the case of late blindness. So the sooner you do this, the more intensive the intervention is, the more you can recruit the visual brain to do that. Right? I'm going to show you some other examples about this. Here's sort of an extreme example, because this is an interesting experiment. When I was a fellow, when I first joined the group, this was an experiment that I was involved with called the blindfold experiment. So the question was, if you weren't born blind and you weren't a proficient Braille reader, could you turn your visual brain into something else? And this is how it was done. So we took normally sighted adults in their 20s and their 30s, and they came and lived at the hospital for 5 days, and they were blindfolded throughout that period. And every day they were professionally taught Braille by a professional Braille instructor for 4 to 6 hours. They lived at the hospital, they hung out at the hospital, and all they did was learn Braille, right? Paid very well. I know what you're thinking, right? New England winters, people are looking for things to do, right? So what you're looking here, what I'm going to show you here is the serial fMRI of the back of their brain as the week proceeds. All right? So on day 1, we put them in the scanner and look at the back of the brain, very, very quiet in terms of response to tactile stimulation. Day 2, you see the visual brain turning more and more tactile. Day 5, it's now robustly activated in response to tactile stimulation. So 5 days is enough to make these changes happen in an adult brain that already has fully normal developed vision. Why do I think this is possible? It's the intensity of the intervention. The profound blindness, the intensity of the training The brain likes that. That's when changes typically occur. A couple of other pieces of information I'll share with you. On the 6th day, we take the blindfold off for 24 hours, and we put them back into the scanner, use the same tactile stimulation, and the activity is very gone. So very, very reversible as well. Other interesting piece that comes to this, we had a control group. So we had a group of individuals who were also at the hospital learning Braille, but they weren't blindfolded. So they were going through the same formal instruction, right? These individuals did not learn Braille as quickly and as well as the individuals who were blindfolded, right? So the blindfolding seemed to have been able to accelerate their Braille learning ability. The other thing that's interesting is when we take the blindfold off, we give them 24 hours and we test them again, what do you think happened to their Braille reading skill? Went down, exactly. So putting vision back into the equation threw the system out as well. So I think it's an interesting question. If you have an individual who you know is going to go blind from RP, for example, should you teach them Braille through sight, the residual vision, or should you teach them blindfold? I think it's an interesting impact along those lines. Here's the group data, just to show you that it wasn't just one individual. We had over 36 individuals enrolled in this study. I'm showing you what's called a two-way comparison. I take all the activity of the brain on day 5. I subtract that from day 1. And then I take all the individuals in the blindfold group versus all the individuals in the control group. I subtract all that out, and I show you where the activation is. And sure enough, that activity is right in primary visual cortex. So all of this is happening across the groups in this particular part of the brain. Let me show you the opposite situation. So this is something called Project Prakash. This is led by a colleague of mine, a dear friend named Pavan Sinha from MIT. He's of Indian origin, and one day while visiting his native country of India said, you know, it's absolutely astonishing of the number of blind children in this country, and they have the largest number per capita of blind children in the world. And the other thing that's pretty astonishing is the majority of them have treatable blindness. In most cases, it's cataracts. So he had a very, very interesting question. He said, you know, what if we had the chance to learn how we see? Right? It's very, very hard to ask a baby what they're going through, obviously, as they learn to see. But what if you had the opportunity of taking a young adult, giving them sight, and asking them what it's like to learn to see? And that's exactly what his project is. Project Prakash, Prakash means light. So he's essentially giving light to these children. And it's a wonderful, wonderful project because not only is he trying to answer a very, very important neuroscientific question, he's also trying to have this really strong humanitarian arm as well. Close to now 30,000 children screened, over close now to 2,000 who have been provided free cataract surgery. It's really quite remarkable. I'm going to show you a video from one of the children. This is Sumitra. She's 12 years old. She had dense cataracts due to rubella as a child, and I'm going to show you the baseline situation and a very, very simple scenario to kind of show you what's happening here. So here's Sumitra pre-surgery. She's walking in, again, as I said, she's 12 years old, and they put a box of chocolates right next to the wall there on her left, and we ask her, Sumitra, can you find the box of chocolates? And she looks around, has no idea, and this is a classic sort of scenario. They feel really kind of like they're on the spot, and they feel like they're failing, and they just sort of like hunker down like this, and they get very, very discouraged in these types of situations like this. Despite probing, despite some indications and prompting, she really has no idea where the chocolates are. So she's going to get up here. And this is about 3 to 4 days after cataract surgery. Not years, not months, 3-4 days. Bring her back into the corridor, and without any prompting, there you go, grabs the box of chocolates right away. So really, really dramatic. When you think in a country like India with extreme poverty, extreme issues, this is monumental. And it's really, really life-changing for these individuals as well. So it's an incredible opportunity, as I said, to understand visual development, but at the same time also there's this incredible humanitarian arm as well. So we have a study now with Pavan where they're scanning these kids, doing the MRIs, and shipping us the MRIs through the internet, and we're looking at their brains as well. And what I'm showing you in this particular photo down here is we're comparing all the kids who got cataract surgery versus the kids who didn't. And the biggest changes in terms of cortical thickness, and thickness is an indicator of brain development, is right again in the primary visual cortex. Right? So again, this is where things are happening. Notice the location here, how similar it is in this particular location here. So the biggest changes seem to happen in the primary visual cortex in this particular case. Now I know what you're thinking. Okay, you're coming in with all your fancy brain scans, and how do I know this isn't Photoshop and this sort of thing? I'm going to give you some experimental evidence that indeed what's happening in the visual cortex actually matters in this particular case. So this is a clinical case that a patient I should say, I had the chance to work with, but this story happened before I came to the hospital. So I'm telling you the story the way that I was told the story. In the particular case, at the time of the study, this was a 63-year-old right-handed female. She was blind at birth. She had retinopathy of prematurity, profound because her reported visual acuity was no light perception in either eye. She hits normal milestones. She learns to read Braille at the age of 6, and she's a highly, highly proficient Braille reader. I don't need to convince you that 120, 150 symbols per minute is a very, very fast Braille reading speed. This is someone who is very, very good at this skill. In fact, she was an editor for a Braille journal back in Spain for La Once, which is the Spanish organization for the blind. Very, very proficient Braille reader. One day, she's not feeling well. She wakes up, she has a really, really bad headache, she has difficulty swallowing, tingling sensations. She doesn't really know what's going on, but she decides to go to work nonetheless. A few hours later, symptoms get worse. She passes out, she loses consciousness. She's rushed to the hospital and she falls into a coma for 24 hours. She wakes up, the doctors come in and say, you're fine, you're safe, you had an event, everything's okay, everything's going to be all right. And she said, great. Thank you so much. Would you mind giving me my phone book, my personal effects, because I'd like to contact some family members and so on. They said, of course. They hand her her phone book, which is written in Braille, and she starts reading it like this, and she says, you know, this is really, really strange. I can feel the dots, but I have no idea what this says. Anybody know what happened? I'll give you a hint. She had a stroke. Question is where? In her visual cortex, right? So it was an embolism at the tip of the basilar artery, knocked out both banks of the occipital circulation, and to this day she is alexic, the acquired inability to read Braille because of damage to her visual brain. Not the part of the brain responsible for language, not the part of the brain responsible for touch, the part of the brain that theoretically she wouldn't have use for, right? So pretty pervasive, pretty tragic evidence that indeed what's happening in the visual cortex is intimately related to these compensatory behaviors. Let me give you more experimental evidence for this, and this was also part of the work that I did as a fellow. It turns out there's a way to stimulate the brain using a technique called transcranial magnetic stimulation, non-invasively, and I'll explain to you very quickly how this works. So TMS is essentially a big battery. It's a machine that stores an enormous amount of charge, and when you hold this coil over a person's skull, you can actually send a magnetic pulse, an electromagnetic pulse through the brain that can stimulate or inhibit the brain below it, depending on the frequency that you use. So if I use a low frequency, so sending one pulse about every second, I can silence the brain. I can deactivate the brain. If I stimulate at high frequency, around 10 to 20 hertz, 10 to 20 pulses per second, I can actually excite the brain. So this is what we did working with proficient Braille readers. So what you see here, what's called the sham condition, in other words, we just hold the coil and there's no stimulation. They're reading at about 70 symbols, 70 words per minute. When we use low-frequency stimulation, we can decrease their Braille reading speed, right? So we're silencing the visual brain. When we use high-frequency stimulation, we increase their Braille reading speed. And we do this in a randomized fashion, so they don't really know what's coming, and the results really seem to hold true. So this is active manipulation of their visual cortex that is impacting their ability for Braille reading. So again, showing you causal evidence that what's happening at the level of the visual brain is intimately related to their compensatory behaviors. So here's my summary slide in terms of ocular blindness. In the setting of ocular blindness, compensatory skills and behaviors are intimately linked to how the brain rewires itself, and in particular, the developmental fate of the occipital visual cortex. So here's a summary visual of those same principles. In the case of ocular blindness, the visual brain becomes non-visual. It is responsive and is responsible for the processing of touch, for memory, for smell, all sorts of language skills. And this, again, as I said, is the compensatory seat of behaviors, or the seat of compensatory behavior, I should say, in the case of ocular blindness. So now I think this begs the question, what happens if you're born with your eyes and they're fine, but your occipital cortex is damaged? Where does all this compensation happen, or does it even happen at all? And sure enough, there's a population that exactly fits very much that profile, and that's CVI, of course, or the case of cortical or cerebral visual impairment. And you're all very, very familiar with this. So let me just give you some background about this, so we're all on the same page. So CVI affects nearly 2 out of every 1,000 live births, and accounts for nearly 20 to 25% of visually impaired children in developed countries, not developing, developed countries. And I was really, really struck. When I was going through school, I had never heard of CVI. I had no idea what CVI was. And now, all of a sudden, it's this public health crisis. So this is some epidemiology data that comes from the United States that rank orders all the causes I should say, the rank order of causes of visual impairment in children in schools for the blind throughout the country. And number one is cortical visual impairment. And it's really striking. When I talk to ophthalmologists and optometrists back in Boston, I ask them this question: where do you think CVI ranks? They have no idea that it's a top cause, in fact, the top cause. Look at the other things just out of curiosity. You have things, for example, like syphilis and herpes, right? These are all things that are infectious, right? Theoretically, we'll get better and better at taking care of this. Retinopathy of prematurity has always been a big one, but we figured that out. We figured out that we were the ones doing that, and we're much better at taking care of these children, right? And then you also have to think that as we get better with things like retinitis pigmentosa and Leber's, genetic gene therapy, gene studies, and so on, that's also going to decrease as well. So I submit to you that over 20, 30, 40, 50 years, the profile of what it means to be visually impaired is going to change dramatically as we get better and better dealing with these causes, but not so good in terms of CVI. So I think we're heading into a very dramatic situation. The question is why are they so prevalent? Why are all these kids out there? And of course, the reason is we're getting very, very good at taking care of premature babies. We weren't so good before, right? 24 weeks, 26 weeks is not unheard of now to survive. And the problem is they're surviving with complications, right? And CVI is a big one. A little bit more information about this. The major cause of CVI, again, as you know, perinatal hypoxia, ischemia, is the biggest driver. There is a bleed that happens in utero. There's blood that rushes into the ventricles of the brain. And as the baby develops, this blood is resorbed, but you see now this enlargement of the ventricles, and there is what's called focal necrosis of all the white matter pathways that surround the ventricle. I'll get back to that in just a second. Head injury, trauma, also possibilities. Infection like encephalitis is another one, but the big one is certainly some sort of hypoxic or ischemic event. Other things to think about in the case of CVI. Typically, we say that these kids with CVI have a normal eye examination. That's not entirely true. I think a better way to think about it is that they have visual disturbances or visual dysfunction that can't be explained by their eyes, right? It's not a refractive error, it's not strabismus, it's not an infection or anything along those lines. There are higher order complex visual processing problems that can't be explained by the eyes, and that's what makes it very, very challenging. They go and see their eye doctor, and said, looks okay to me, your kid must be faking this, your kid must have some sort of learning disability, your kid must be lazy. I'm sure you've all heard similar stories as well, and this is what concerns me tremendously, not just on the clinical side, but also on the care side as well. Visual acuity can range from anywhere from near normal to profound blindness. Visual field deficits are also very, very common, and typically they're in the inferior visual field. And I'll give you some neuroimaging evidence as to why that's the case. And the characteristic finding in a lot of these kids is what's called PVL, periventricular leukomalacia, right? So periventricular meaning surrounding the ventricles, leuco meaning white, and malacia meaning soft. Right? And it's not because somebody took their brain, stuck their finger and said, oh, that's soft. No, it's actually because it looks soft on an MRI. That area has a sort of a fluffy appearance, and that's what PVL comes from. Typically, they have a medical history with that includes some sort of neurological impairment. Typically, it's cerebral palsy when it's affecting motor pathways as well, cortical spinal tract, so often you see the two together. And in the presence also of unique visual behavioral dysfunctions, as I said, including visuospatial and motion processing, complexity, crowding, and attention deficits. These aren't things you can fix with glasses, right? These are things that are higher order. They're definitely somehow involved with how the brain processes information. Some background as well that you might be familiar with. A lot of individuals in this field have called this, or have referred to this as a dorsal stream dysfunction. So just to remind you that the visual system is parsed into three major streams. There is what's called the dorsal pathway. This is the where pathway. This connects the occipital cortex to the parietal cortex and to the front of the brain. This is responsible for identifying where things are in space, right? There is also the ventral stream, or the ventral pathway. This connects the occipital cortex to the temporal cortex. This is involved with identifying an object, the actual identity of an object. And these two things, there's a division of labor between these two aspects. And the third pathway, which represents direct connections from the occipital cortex to the frontal cortex, this is responsible for attention and eye movements. And this division of labor exists in the visual system. And we also know that clinically this is true because damage to one stream spares the others. And the idea now is CVI a dorsal stream dysfunction? Right? That's a big sort of theory. And we wanted to get into this idea in terms of neuroimaging to try to give a neurophysiological basis for it. Just some other pieces of information. Gordon Dutton, you might know this individual, did a lot of work as well, has an interesting perspective called the tree of visual perception. He talks about how eyes are kind of like the roots, and then there's one big branch, which is the ventral stream. The other big branch is the dorsal stream. I think it's kind of an interesting way to think about it. I'll be honest with you, I'm not a big fan of this particular view because I think there's a misconception that vision is just a bottom-up phenomenon, that it comes into the eyes and is somehow magically is all figured out in the brain. The fact of the matter is that the brain is really a loop. It looks more like this in terms of information is being constantly cycled all the time. What's coming in versus what you think you see is constantly being compared. And I think in the case of CVI, there is disruption. And in particular, I think there's a problem of connectivity in the brain as well. And we'll get deeper and deeper into this issue as well. So let's start looking at some data. I'm showing you here standard MRI images, right? If you were to go and ask for an MRI of one of your patients, this is what it would look like. This is an axial scan slicing through the brain. Here is a sighted control individual in their late teens. Here is an ocular blind individual. Looks awfully similar, doesn't it, right? Here are two individuals with CVI that we've been working with. This one, CVI1, is a 16-year-old female. CVI2 is a 22-year-old male. And the first thing you see is this enlarged ventricle form here, right? These enlarged ventricles, again, what we call PVL, periventricular malacia. The problem is we can't really tell more than that, apart from the fact that they have PVL. In other words, standard imaging doesn't tell us anything about their underlying deficits, right? And I'm going to try to demonstrate that for you in this particular game. We're going to play a game here, right? You guys ready? You guys remember Sesame Street? Right? One of these brains is not like the other, right? We're going to do that right now. So I have 4 brains here that I'm going to show you. And you can all see they have different levels in terms of their ventricular structure, right? So pretty extreme here, a little bit intermediate, and looking pretty normal there. So my question to you now is, who has the best acuity and who has the worst visual acuity, just based on what you see? And just to, for the interest of time, your rationale probably said something like this. You said this is probably the most normal looking brain there, so that's probably going to be the best acuity. This one's probably intermediate, number 2, and it's probably a toss-up between number 3 and number 1. Maybe number 1 because it seems a little bit more symmetrical there, right? Does that kind of make sense? How you'd think? Well, here are the answers. It turns out that the worst visual acuity was number 2. Number 3 actually has normal visual acuity. The intermediate one was number 1, and number 4 is actually in a coma. I just threw that in. We had no idea what... Just to show you, or to make my point, I should say, what we call the neuroradiology paradox. And the fact is that lesion load, the extent of apparent damage, does not always correlate with clinical symptoms. And that's the problem. I see this in my clinical practice as well. Sometimes I look at an MRI, there's extensive damage, and I say, wow, this guy is really, really in bad shape. I go into the room and he's standing up talking to me. Right? There's other ones I've seen where I'm like, yeah, that looks fine. I don't see anything. And sure enough, you know, they can't speak or they have all sorts of motor issues as well. And that is the big challenge, particularly in the case of CVI, is that even with MRI imaging, it doesn't really tell us what's going on with these kids and with these individuals. So we need another way to scan them. And let's get into that right now. So here's my hypothesis that I hope to kind of convince you. CVI is a disorder of brain connectivity. Underlying connectivity is associated with observed visual dysfunctions, and this is how we're going to try to figure this out. So we use a technique called diffusion-based imaging. Right? This is not standard MRI. This is a different way of looking at the brain, and allows us to figure out the brain connectivity, the white matter projections through the brain. And this is how it works. So it turns out that your brain is essentially anywhere between 70 to 90% water. Right? So what the scanner is doing is tracking the motion of water in your brain. And if it was following one water molecule and it was moving equally in every direction in 3-dimensional space, we would call that isotropic diffusion. So in other words, no matter where it was, it could move freely. We call that isotropic diffusion. If, however, I'm talking a little bit quicker than the video, if, however, that motion was somehow constrained, in other words, it was moving along one axis more than it was along the other axes, we call this anisotropic diffusion. And why would that water movement be constrained? Well, if it was associated with a brain cell, right? So that axon constrains the motion of that water molecule. And what the scanner is doing is figuring out voxel by voxel, pixel by pixel, what those water molecules are doing, and from there inferring the orientation of white matter tracks. The nice thing about this is that you can do something what's called virtual dissection. You can pull tracks out in terms of looking at specific areas and connectivity and parts of the brain to see how they're wired together. Here's a nice example looking just at the optic tracts, for example, from the thalamus in the middle here all the way to the back of the brain. These are the optic radiations. So it allows you to reconstruct specific tracts of interest. So here in this video is an example of what this technique looks like. And I'm going to rotate this in 3 dimensions. This is a 3-dimensional reconstruction of a 15-year-old normal-sided control of their brain. And here's the dissection that I talked to you about. So what we did here is we asked the software, tell us how the visual brain in the back here is connected to the rest of the brain. All right? So we can look specifically at the information superhighway, if you will, of your brain. This is the connectivity of your brain. A couple things to keep in mind. First of all, the brain is not pretty colors like this, obviously. That's just a nomenclature system. Blue means it's axons that are traveling up and down. Green meaning to the front and to the back of the brain. And red means moving side to side. Right? So it's just an orientation. The second thing is it doesn't look like spaghetti like this either. That's just the resolution of the software. Each one of those thin threads is thousands and thousands of axons of brain cells, and it just has to do with the resolution ability of that. So just because you don't see the axon doesn't mean there's nothing. It just means that there's a really, really small number. All right? So let's take a look in the case of CVI, what happens, right? So here's again a normally sided control. We've asked the software to just show us the brain connectivity of the visual cortex to the rest of the brain, and there's 3 main pathways that show up. And the names aren't really important. The SLF, the IFOF, and the ILF. So you should remember back a couple slides I told you about the 3 pathways of the visual brain. This is the neuroanatomical correlate of those 3 pathways. The SLF is the dorsal stream, the ILF is the ventral stream, and the IFOF is the direct connections between the occipital cortex and the frontal cortex. So we now have the neuroanatomy of the 3 visual pathways, right? Anybody want to know what a CVI individual looks like? Here it is. Yeah, a lot missing. Yeah, it's pretty dramatic, age matched. Okay? And some of the things to think about this particular individual. We noticed that in this particular individual, she was quite good at identifying objects. In other words, her ventral stream seemed to be largely intact. She also had a lot of deficits in terms of her spatial processing, and sure enough, her SLF seems quite thin. But her biggest deficit was attention. And sure enough, we can't even reconstruct her IFOF pathways as well. So this gets back to this idea that the connectivity should somehow reflect the deficits that these individuals are showing. Okay? So let's get a little bit deeper into this point here. You're looking at the left hemisphere here. Again, those 3 same pathways, right? The ILF, the IOF, the SLF, as I told you. This is a normally sided control. Here's an ocular blind individual of the same age. What do you notice? It's all there, right? In other words, all the machinery is there, right? Very, very similar to what it looks like in a normal sided control. And here are those 2 CVI individuals I mentioned. Again, a lot of thinning, a lot of pathways seem to be very, very thin. Right? In some cases, the ILF seems to be quite nice, the ventral stream, but the dorsal stream seems to be showing the most deficit. So kind of fitting with this idea of spatial deficits and whether or not they can make sense by underlying connectivity. So we want to pursue that a little bit more. Another thing, just to give you some fancy math, we can also do what's called network analysis. We can look at the brain and figure out and count the number of connections between various areas. All right? So we use what's called network-based statistics. Here it is. In the case of a sided control, these are all the connections between the brain. You're looking at the brain from the top. Here it is in an ocular blind individual. It is hyper-connected. Right? That seems to make sense given all these compensatory behaviors. By enhancing connections between non-visual areas and eventually the visual cortex, this supports these compensatory behaviors. So we're not the first ones to show this. There are many groups who have shown this in the past as well. But the dramatic thing is the case of CVI. It's an under-connected brain. Right, compared to the case of ocular blindness in the case of sided controls as well. Just more math behind this. This is called the connectivity matrix. This allows you to quantify the number of connections between various areas by hemisphere, by brain type, and so on. The more red you have, the more connectivity you have. Notice that there's a lot more red in the case of ocular blindness and a lot less red in the case of CVI as well. So ocular blindness, hyper-connected, CVI, under-connected. All right? Other things to think about. I mentioned this idea about cortical thickness, how we look at primary visual cortex as a site of brain development, giving us a sense of what's going on, right? I gave you that example of blindfold subjects, and also in the case of the Project Prakash. Here it is in the case of sided controls. Red means lots of thickness. That's good. The brighter it is, the thicker it is. Case of sided controls, very, very thick. Case of ocular blindness, also very, very thick. Here it is in the case of CVI, very, very thin. Moral of the story is that this is a very underdeveloped visual brain. right? And it goes throughout life this way. That's the problem. So the question is, how do you kickstart this? How do you get this to start growing, if you will, so that the rest of the brain grows as well? And that's a really big, important question. Our approach is the following. I'm now going to give you a little bit more evidence behind that. I spent some time in terms of, you all know the clinical deficits. I gave you some evidence from the structural standpoint, the brain wiring. Now we're going to talk a little bit more about the functional components of this. How does this relate to the actual behavioral deficits that we see in these individuals? And this is how we started doing it. So as you know, it's a challenge working with these individuals. The attention isn't always there. Fixation is also kind of tough to do. They're always, kind of not that interested in what you're doing. The last thing they want to do is to go through an eye exam. So we spent a lot of time developing tablet-based and tactile-based eye examinations and visual assessments where the kids are very, very engaged. They hold the tablet themselves. If they're nonverbal, they can touch and tell us what they see, what they don't see. And we also use a lot of artificial intelligence behind this, what's called Bayesian approaches, where your first answer ultimately affects the next question you get asked. And this allows you to get to the answer much quicker that way. So the tests are much shorter in this particular approach. Some people have demonstrated changes in contrast sensitivity, for example, in these kids. That I think is an interesting phenomenon. We haven't seen huge deficits with this. The problem also with contrast is that it's hard to test. It takes a long time to do it. Some interesting also data in terms of biological motion, right? One of the biggest things that you hear in these kids is they say, I can't recognize my parents in a crowd. I can't walk and understand what's around me, right? Or I really don't like watching TV programs where there's a lot of action. I prefer things that are much calmer, much stiller. And there's some thought that they're really not able to integrate biological motion or understand complex motion as well. And we saw a little bit of evidence of that. So we wanted to go a little bit deeper into this motion integration question. And this is how we did it. So there's a lot of work in the psychophysical world about trying to understand motion integration. J.J. Gibson was a famous psychophysicist, and he studied how pilots would land their planes, and their understanding of what's called optic flow, how you move through the environment and how information moves around you, is a really part and parcel property of the visual brain. Its ability to integrate all that motion around you in terms of your heading, either moving forward or backwards. And we tried to replicate that using a very, very simple visual stimulus, which I'll show you here. Again, this is called optic flow. So I'll ask you to look at this visual stimulus here, and you'll notice these dots are going to start moving, right, kind of randomly. And then at one point, you'll see they're expanding, and then they're going to start going inwards, and they're going to come out again, and they're going to go inwards. Everybody see that, yeah? All right. The interesting thing about this is that this is a replication of optic flow. As you move in and out of an environment, this is how information moves around you. The other thing about this is that the number of dots that have to move together is a measure of your sensitivity. In other words, how much information do you need in order to know that you're moving forward versus backwards, right? And that's called motion coherence. The more dots that have to move together, the more impaired you are in this ability. If you only need a small percentage of the dots, that means that you are able to move through the environment and make sense of what's happening around you. All right? There are other advantages by using the stimulus as well. So as I said, there's the ecological relevance of moving through an environment. Notice also that it's very, very easily quantifiable. We can measure the coherence threshold, as I mentioned, by the number of dots that move. Notice also that if you're not looking at the center, you can still tell me what direction it's moving in. So even if they have poor fixation, we still can get the data. Notice that those of you sitting in the front versus those sitting in the back also see it moving back and forth. And it turns out that it's also invariant by viewing distance as well. So even if the child is like, we still can get the data pretty reliable. So that's what makes this quite robust in terms of our ability to measure it. So let's take a look at some of that data. So here it is. This is motion coherence thresholds. Like remember, the worse your threshold is, the more impaired you are at being able to do this task, all right? So normal thresholds are somewhere between 5 and 25%. In other words, 5 to 25% of the dots have to move together in order for you to be able to tell me what direction you're moving in, forwards or backwards. And here's our group of age-matched controls, about 18 kids, and the threshold was about 16. So right in the middle of what the group population data is in the literature. And here are our CVI kids, about 3 times worse. In other words, they need 3 times the information in order to be able to tell you what direction things are moving in. So this, again, behaviorally is an indication of an impaired processing system, right? Let's go back to the neuroscience question. Reminding you again that the 2 streams, the dorsal stream and the ventral stream, the where and the what pathway, all have different modules responsible for different things, color, faces, things like that. And it turns out that there's a part of the brain, sorry, it's a little bit covered here, called area MT. It stands for middle temporal. This is the part of the brain responsible for motion integration. That's all it does. It's only interested in moving objects. And notice that it's sitting in the dorsal stream. So now we have a surrogate, a functional surrogate for dorsal stream processing. Right? So let's take a look. Let's put these kids in the fMRI scanner and see what happens. Here's a normally sighted, normal developed, age-matched control. This hot spot of yellow that you see here is area MT. The yellow circle around there is just a probability map showing you where MT should be. And sure enough, it's very, very strongly active. Again, using that motion stimulus that I showed you. Here's our CVI subject. Basically nothing, right? Really, really a smattering. To get anywhere close to this level of activation, they need basically 100% coherence. In other words, all the dots have to move together one way to the other to get MT to even respond. So again, a really underperforming and underdeveloped visual system in this particular area. The connectivity also doesn't seem to be right as well. If we look at the connectivity between primary visual cortex and area MT, here's our normally sighted control, lots of arborization, pathways going all over the place, a lot shorter, a lot more compact in the case of CVI as well, and we can quantify this. So the moral of the story is we can try to put this all together. We have their visual deficit, we have their structural anatomy, and we also have their functional anatomy, and we think we can piece all this together. So that was the motion perception story. Let me give you another piece of evidence in terms of visual fields, which is also a very, very common deficit or common complaint in these children as well. So you're all familiar with perimetry, right? Like the Humphrey visual perimetry and so on. It allows you, you look at the dots and it gives you a visual field representation. This is exactly the same thing, so we're going to do it on the surface of the visual brain. Right? So we can map the visual world, on the surface of the visual cortex. And this is how we do it. A technique called retinotopy. Notice, for example, as this ring expands, notice how the signal propagates over the surface of the visual cortex. Notice on the figure on the right, when I stimulate the left side of the visual field, I get activation in the right hemisphere. All the warm colors, which is the upper visual field, show up in the lower bank. All the cold colors, the lower visual field, end up on the upper bank. So that's how we know the visual brain is organized. Left is right, right is left, up is down, down is up. And this is just a recreation, a remapping of that on the visual surface, the surface of the visual brain. So let's take a look. Again, here's our normal sighted control. We did a very, very simple experiment. We drove the upper visual field in yellow, the lower visual field in blue. And notice in our normal sighted control, lots of blue up top here, lots of yellow down here for the upper and lower visual field. In our CVI subject, you start seeing these gaps, right? There, and this large one there, and particularly corresponding to the lower visual field, right? So what would you guess your visual field would look like? Where would the deficit be? Yeah, here's your Humphrey. Yeah, that's exactly right. So there's an immediate correspondence of where the brain is activated in these early visual areas in terms of where the visual field deficit is. And this is just a mathematical a computation, I should say, of the strength of activation. The lowest activation is in this lower visual field. We can also look at the connectivity of this as well, the optic radiations, as you all know. The upper bank is responsible for the lower visual field. The lower bank the lower bank, I should say, for the upper visual field, the upper bank responsible for the lower visual field. Nice thick arborization in the case of this normal sided control. Here's our CVI subject. Essentially, the lower field is missing, right? So again, the function, the structure, and the visual deficits all fit together. That's kind of the idea, and that's very, very much the goal. So our thought is that we should be able to look at any measurement of visual function, give you why the structural connectivity isn't there, related to the structural connectivity, and at the same time with functional imaging tell you why the brain doesn't activate correctly. And no matter what way you enter the system, if I know one piece, I should be able to predict the other two. And that's kind of the idea of how the wheel turns. And as we go through more and more types of visual assessments, we'll get a bigger and better picture of what's going on in terms of the underlying physiology of this condition. So my summary slide for this particular case is that in the setting of CVI, there appears to be extensive reorganization throughout the brain. However, how this rewiring relates to visual development, compensatory strategies, and the recovery of function remains largely unknown. Very, very different than the situation I presented in the case of ocular blindness, right? Does this at all surprise you that strategies that you develop for ocular blind don't work in CVI and vice versa? Their brains are fundamentally different, right? But how do they come to you? Visual acuity. Right? That's the problem, right? We classify them using the same sort of criteria, but fundamentally these are two very, very different scenarios, and I hope I've convinced you of that. couple of other comments I want to share with you. A lot of people ask us, how are we able to scan these kids, right? Because we don't anesthetize them. A lot of the past studies, for example, have anesthetized these children. The problem is you can't do functional imaging in a child who's asleep, right? They have to be awake. That's how we get the data. Just a couple of things that I want to share with you. First and foremost, we spend a lot of time working with these kids, getting to know them, building their trust, not only with the kids, but also with the family. We have a mock scanner where we do the work. So it looks exactly the same as the real scanner, but there's no active magnet. So they can go in, they can climb around, they can touch anything they want. It's not a problem. There's no security concern or safety concern for them. We also tell them to bring their favorite stuffed animals, and we scan them too, and then we subtract them out in terms of the signal as well, so that they're comfortable. We ask the mom, for example, in this particular case, to be involved, and she's very, very close and reassuring, so that the parents are always, big team members in this work as well. And as I said, we really try to get to know what works and what doesn't work. We have MP3 files of the scanner noises, which we send to the family, and the kid listens to it in their iPod before they come, so they get a sense of what the sounds are like also. A lot of prep time to try to understand what these kids are going through. Last thing I'll mention, we try to figure out, as I said, what works and what doesn't. This particular child, interestingly enough, for whatever reason, loves Egyptology. She watches all sorts of movies and cartoons and reads books and so on, and we said, oh, that's great. So we're going to wrap you up in a blanket now, right, nice and tight, just like the mummy. Here's the mummy going into the sarcophagus. She didn't budge, not even a millimeter. Absolutely incredible. We have stable data on these individuals because we work very, very closely with them. We really don't have this idea, and I think in the past people said, oh, let's just put them in and see what happens. No, you have to build a rapport with these individuals, and I think that's what allows us to get higher quality data than past studies. Another one here, this isn't our scanning bay, just to show you the creativity that a lot of people are developing. This is one site, I think in Milwaukee, if I'm not mistaken. And they've had this whole yellow submarine, and we're watching, and you're a scuba diver, you're going through, and all this sort of thing. And when the kids are engaged, I know I'm preaching to the choir. When the kids are engaged, when there's fun, and when they're safe, when they feel confident, that's when good things happen. And I think that's a very, very important piece to the study as well. Some final thoughts as I wrap up here. Some of you might remember when Kevin Costner made good movies, a long, long time ago. Yeah. And there was one particular called Field of Dreams, where he played this Iowa farmer who hears these voices in his head, build it and they will come, build it and they will come. And he gets the bright idea to just mow down half his cornfield and build a baseball stadium. And I think at some point his dad shows up and I fell asleep. But that was the point of the story. And I'll be very honest with you, when I started this project, that's very, very much what it was like. I was going to conferences like AAR and so on, talking about ocular blindness. And there would be voices in the crowd, what about CVI, CVI, CVI? And the more I thought about it, the more I thought that, yeah, there was a really incredible opportunity here, and that's how the project started. The concern that I have is that, will this be a field of dreams? I can tell you that there are some parents, for example, who just simply don't want to know. Thanks, but no thanks. You're not treating my kid. I've gone through 15, 20 years of this. You're not going to add anything to their life. So I really hope it doesn't turn into a field of dreams type situation. We really hope that this can indeed make an impact and offer some neurophysiological basis to something that obviously is very, very important to all of us. Let me give you sort of a scenario that challenges us. Let's say we pull this study off. We're able to demonstrate, for example, that it is indeed dorsal stream dysfunction and demonstrate the neurophysiology of this condition. So if it is indeed a dorsal stream dysfunction, now what? Now what do? Does that mean that you need to train more spatial tasks on these kids to get that up to speed? Or do you use more visual identification tasks to try to bootstrap the spatial components, right? I don't think anybody knows, but I think we do have the opportunity to try to look at this systematically. I think that's really, I think, what the field needs and what it hasn't had in the past. I always use the analogy of autism, for example. It wasn't that long ago that we thought autism was caused by mothers who neglected their children, what we used to call the refrigerator mother principle. This was in medical records, right? Mothers who were abandoning their children. Those of you who have any experience in this area know that could anything be farther from the truth, right? We were dead wrong about this. And it took a mass effort of concerned parents, educators, celebrities in some cases, politicians, scientists, all coming together and applying enough pressure from enough directions to create an upswell movement to finally change the way that these kids were taught and taken care of. And I think CVI deserves exactly the same thing. Some final thoughts and some potential study impact, and this leads to my final slide here. Evidence-based approaches, dispelling neuromyths. I think that's one thing that neuroscience can bring to this area. What works, what doesn't, and why, right? Identification of neural correlates, structural and functional associated with visual dysfunction. For example, is it indeed dorsal dysfunction? In my experience, it's not that simple. Some kids have both. They're not mutually exclusive, for example. But does the physiology fit the visual deficits? Targets for deficit-based training. As we get better and better at assessing these kids beyond visual fields, beyond visual acuity, we'll have strategies and targets that we should work on to try to enhance their vision, wake up this visual cortex, as I said. Longitudinal tracking of development. That is the ultimate goal. You can imagine these kids getting scanned at baseline and 5, 6, 7 years throughout school, scanning them afterwards and trying to parse those kids who do well versus those kids who don't, and trying to understand why from a brain anatomy standpoint. And I think the last piece, which is the most important, is the community empowerment aspect as well, which is what I think CVI really, really deserves. We have a Facebook page. I promise you I get more traffic on this than I do my emails. It's really incredible. We post stories, kids that are participants and parents who are involved post as well, relevant stories. I invite you to come and like us if you are interested. We're the CVI Neuroplasticity Research Group. This has been a big piece in trying to get the community engaged and involved with this. Last story, second to last story I'll share with you. I'll give you an example of what I think is immediate impact. When we first published our initial findings about this tractography and visual deficits, as luck would have it, it ended up being the cover figure of this journal, right? We had nothing to do with that. The editor ended up choosing the pretty pictures, if you will. So we thought that was kind of interesting. We asked for reprints, and we started sending it to the kids who were involved in the study. And we said, hey, isn't this kind of cool? Your brain is on the cover of this scientific magazine. One of the kids woke up the next morning, took the magazine, walked into her teacher's office and said, you see, I told you there was something wrong. And she started calling the principal, her doctors, everybody, and saying, I told you there was something fundamentally wrong. I'm not faking it. I don't have some sort of emotional problems or something like that. So there is a sense of empowerment, I think, getting involved with this and saying, maybe not so much ownership, or maybe not so much that there's something wrong in my brain per se, but they feel a little bit more reassured that there's something indeed that can be explained from a rational standpoint as opposed to just being dismissed very quickly. So I think there's something to be said about that. I want to just identify, say thank you in the last minutes that I have, the individuals involved. It's a massive team effort, as you might imagine, to take on a project like this. On the clinical side, we have Dr. Jenna Hadari. She's a neuropediatric ophthalmologist from Children's Hospital of Boston. I think in this particular case, you have to align yourself with clinicians who even know what CVI is, first of all, for starters. That's a big piece. Unfortunately, we have someone who can do that. Peter Becks is a psychophysicist. He develops all the visual tablet-based testing for us. And what I call the Three Musketeers, this is the group at Perkins School for the Blind. Derek Wright, an OTM specialist, Dr. Barry Cran, an optometrist, the head of the clinic, and Louisa Meyer, who's involved also for the evaluations and perimetry. These people tell us the reality. They work with the Perkins kids. They tell us every day what they're struggling with, what's a concern, what their parents are concerned about. So this is where we get our real world information. On the structural side, I have Karina Bauer, who's a postdoctoral fellow in the lab. She brought a lot of that expertise in terms of the brain reconstruction. The handsome gentleman next to her is Andy Ellison. He runs the scanner, so that worked out really well. And they actually got married last month, so I keep telling them, please don't get in a fight. This is a really important study. And on the functional neuroimaging side, David Summers, a professor at BU, Boston University. When I did my one of my fellowships I did with this individual, everything I learned from brain imaging, I learned from him, so it's great to bring him back on board. Gabriella Hirsch is a research assistant in the lab and really took on the fMRI work, and I really challenged her a year ago if she could come up with the analysis methodology and so on. So a really wonderful team, and it's a privilege to work with these individuals. And like all of you know, good team, good people makes life a lot easier. Thank you again, just identifying the Perkins School for the Blind, Carroll Center for the Blind as well, which I have a close relationship with, Boston University, the NIH, the Mass Lions Research Fund, Deborah Monroe, also a research fund, also our patients and our families. And I want to give a shout out to all of you. You guys are my heroes. Don't ever forget that. Don't ever think that what you do isn't important or doesn't make a difference. My mother is a teacher of preschool children, kids with developmental issues, and I learned very early on the challenges. And I know that she did this because she believed it was the right thing to do, and I'm sure all of you feel exactly the same way. So don't ever forget how important your contribution is to this whole big picture. And the last, I promise, final story I want to share with you is this one here. A great picture. This picture actually is in my office. This is an interesting story. There was a photographer named David Seymour who went around Europe after World War II to try to capture the plight of children after the Second World War and what they were going through. And this particular child was in Italy. He was born blind. learned to read Braille at a very, very young age. And one day this child went out and started playing with something he shouldn't have. It ended up being a landmine. The landmine exploded, destroyed both his hands, and became a double amputee. But he still reads Braille, not because he sees the dots, but because he's using the tip of his nose and the surface of his lips to feel the dots. This, ladies and gentlemen, is neuroplasticity at its best, right? This is what we're trying to understand. When motivation, opportunity, resources, drive are all there, good things happen. And I think you all know that, and I think that's what we really need to be working together with to help these individuals. And with that, I promise I'm done. Thank you.